Good morning, students. Welcome back to Genomics and Proteomics course. And today we'll be discussing Lecture 4, Introduction to Proteomics and Metabolomics. I have divided the lecture into different groups, different sections. One section will discuss about a concept in the first half, more about the proteomics and what is the branch of study and what are the fundamental units of proteins and different aspects of proteins and translation. Then that leads to us to the branch of study of proteomics. And the second part, I will discuss about more on metabolomics, where you will talk more on different metabolites that we have been observed already, primary and secondary metabolites, and the importance of metabolism, catabolism, and anabolism. A little bit more on why the branch of metabolomics exists, and together briefly we will talk about quantitative methods and then disease perspectives of proteomics and the metabolomics. So coming on to first half of the lecture, we are going back into again translation and fundamental units of protein, amino acids. Let's see in the few slides. We have been already discussing in last few classes about the central dogma that already been everyone by now. Everyone we know the classic view: DNA to protein, DNA is transcribed, mRNA is again translated to proteins. Again, when you look in the subcellular localization on the way the phenomenon is happening, in transcription happens in the nucleus, and translation happens in the cytoplasm. Mature mRNA leaves the nucleus and comes to cytoplasm. Then a specific process of translation happens, and a specific polypeptide, chain of a polypeptide, will form up, and then it gives you a specific protein depending on the necessity and the function of the cell. So we have studied already translation in a molecular biology class. I won't go too much deep into that. It should just come back to our basics. So this is an mRNA that's been already translated, a translation in process, in progress. mRNA will have a start codon and then stop codon where translation starts. Based on the signal and then based on the entry point of start codon, a ribosome will come up and then it will start reading a code in the mRNA. Accordingly, it makes a specific amino acid chain and specific polypeptide chain will emerges at the end of. When the stop codon comes here, polymerase leaves the site and then a specific length of polypeptide emerges, and that gives you a specific protein. So we already know that the ribosome will consist of smaller unit and larger subunit, and then these tRNA molecules help in bringing specific amino acids depending on the code here. And then the larger subunit will consist of two sites, A site and P site. A is amino acid transferase tRNA site, P is peptidyl transferase tRNA site. And then the process moves from 5' to 3' direction. And then at the end, a specific polypeptide chain is formed depending on the length of the code and the open reading frame. It should be also noted that once the polymerase leaves the mRNA, same mRNA can be again used to code for the protein, code for the amino acid polypeptide sequence depending on the need and necessity of the cell. It's a newly born polypeptide chain. We already know that how this code in the mRNA will be read. We understood, we already established a concept called codon and anticodon. Codon in 5', 3' of a specific mRNA, specific sequences. And the anticodon comes in the tRNA molecule anticodon loop, and it will recognize the complementary sequences here. And then based on the sequence information available in a triplet codon, it takes an anticodon and brings a specific amino acid. And it should be noted that we already know that the third base within that anticodon is a Wobble base. We already know Wobble position. That it gives rise to degeneracy of the genetic code. We will look into more in the next slide. So this diagram will discuss on more about the RNA code or the genetic code. Just brushing up again our basic concepts. These are the 4 codes. They gives 4 variants of the letters and again 4 more combination, 4 into 4 into 4. We will get into a kind of 64 codons that code for a specific amino acid. That in humans, we already know that only 20 amino acid exist. That's already established in the outer circle here, but they are coded by 64 codons that have been established here. That basically saying that the genetic code, RNA code is degenerate and it is redundant, and at the same time, one amino acid, for example, there is one amino acid that methionine has only one codon AUG, that is a start codon, and tryptophan has only UGG, but other codon, cysteine, it may have more codons. That degeneracy exists here. This we already know, it's an established system. So basically 64 codons code for 20 amino acids. So our system codes for 20 amino acids. The amino acids are nothing but they are the building blocks of proteins in all living organisms. There are over 500 amino acids found in nature so far. So I'm not talking about a single human body. You're talking about all over chemical structure and composition of amino acids. There is a different variants of 500 varieties of amino acids are available. However, the human, we know that 20 amino acids are there. They are divided into essential and non-essential amino acids. That this chart summarizes the both chemical, physical, and other properties of the amino acids here. So you just take an example here, given a chemical structure of amino acid here, what the code letter given to a specific amino acid and the codons it reads. This is a beautifully summarized chart. You can go ahead with that and look into more details in a given time, but I will take an example. Based on chemistry, the amino acids are divided into aliphatic and aromatic. Aliphatic are red circles. Aromatic are kind of, you see, the light green circles here. At the same time, chemical properties, acidic and basic. Based on that, you can divide the amino acids again, different colors. And also one more importantly, the essential and non-essential amino. For example, the name or code letter may be alanine, starting with the initial alphabet A, but it may not be always consistent. For example, lysine will be coded by the letter K, and it is not L. So you should remember that. And when given a sequence of DNA, the amino acids will be given in this code. We will see in one of the slides coming in the lecture. So we already know that the building blocks for proteins are amino acids. The amino acids are read from mRNA in a polypeptide chain. For example, here we are looking at a 3 amino acid polypeptide chain. For example, amino acid 1, amino acid 2, amino acid 3. And when you look into the basic structure of amino acids, you see that there is an amino end, there is a carboxylic end. We call it as N and C end, NC. So usually the polypeptide grows in the direction of N to C. Why is that? Because there is a bonding comes between this hydrogen and hydroxyl group here at the terminal of the C, the beginning of the N in amino acid 1 and amino acid 2. That bond is called peptide bond. One water molecule is removed, one peptide bond bonds, one amino acid added to the other, then again one water molecule removed, then another amino acid adds up. That's called peptide bond that links a polypeptide will form, 2 molecules are moved here. And it should be also noted that it's a basic functional structural unit of the protein, starting with the amino acid polypeptide chain. That's called as primary structure. You can see here. So this is the primary structure. So where a protein consists of a stretch of amino acid, starting with asparagine and then valine here. But the same, you look back into the chemical structure of the amino acids, there is varied chemical composition, varied bonding is there. And it gives, when they come in the vicinity of this variety of amino acids together, they can bind each other by a disulfide bond based on the chemical moieties present. They can have certain structural modifications giving rise to a secondary structure. Yes, sir? It's a primary structure, secondary structure of a protein, where specific components like alpha helix, beta sheets, and helices can form in the structure of the protein. Again, it may go for further on one more conformational chain that's called tertiary structure that gives us a 3-dimensional structure of a specific protein coming from its polypeptide sequence. So this is a tertiary structure when the protein is in isolation. When the protein in a cell is there. But you should remember that the proteins don't work all alone. They need to work in a complex of other protein. There is a complex protein-protein interaction happens. For example, enzyme, apoenzyme. Then you might have a cofactor for the enzyme. Enzyme cannot work alone. So it's a kind of protein complex that will form again a quaternary structure, complex of 2 different proteins or 4 different proteins will form. So starting from a single polypeptide chain, then you need to go along with a specific structural conformation that gives rise to a specific structure and function of a protein. A simple example we have taken from NCBA PubMed, human insulin gene, a transcript variant of mRNA1. So you see that the ATGCs or AUGCs are the mRNA codons. Here sequences here, and the triplet codon forms. When it's translated into protein, basically you look into the amino acid sequence, you read them in alphabetic letters based on the abbreviation of the amino acid here. So this genome sequence, mRNAs, this is the protein sequence or amino acid sequence. So we already know that there is a different structural conformation of the proteins, but not only the sequences coming from the codons to the amino acids, there is another one more functional activity of the proteins you should remember, because each amino acid can be modified again by a specific chemical moiety. For example, here chemical groups or specific proteins or peptides, they undergo a kind of interaction, protein-protein interaction, that gives rise to a specific modification of the protein in addition to the tertiary three-dimensional structure that already been established. For example, endogenous condition, it established there. For example, one moiety that can be acylated, one moiety can be phosphorylated, one moiety, actually this protein is a synthetic protein. You see non-natural amino acids. You can modify the protein to synthetic proteins if you want to make. And there is a kind of fluorophores you can attach, some other chemical moieties you can attach to modify the protein, because you need to detect it at the further studies if you want to do. So a protein after three-dimensional conformation, after forming into tertiary or quaternary also, it can be again modified. That's called post-translational modification. The modification may be either from a chemical group or a proteins or a complex molecules, for example, glycosylation, say sugar moiety that can be attached, isoprenylation, one more example. And as you see here, one fundamental concept you need to remember that, so proteins are structurally different from ribonucleic acid or DNA or RNA. In terms of the fundamental units also, 4 unit nucleotides are there only, but here 20 amino acids are. At the same time, not only a single stretch of molecules you're looking at a conformational change of the protein. So it gives more kind of complexity to decode the protein. For example, you cannot do a PCR, right? You do a PCR for DNA or RNA, but you cannot do a PCR to identify a protein just like starting a primer here and ending here and put it into that. Small amount of protein you put in the reaction, you give rise to tons and tons of molecular daltons of protein. It will not happen because it is a highly complex structural organization, and each residue, there is 20, right? 20 goes into multiplicity, then for example, 100 amino acids here. Then each amino acid, for example, valine here may be acetylated, but valine here may be phosphorylated. So there is a lot of complexity in that. So basically, then you don't do a kind of PCR kind of experiments to study proteins. What you can do is that first isolate the protein from the cell, extract the proteins through some specific protocols. Then you basically use a method called cleavage or proteolysis. Disulfide bonds are there, peptide bonds are there. You breaking at each step, and then you break the conformational chain. You go back into the polypeptide chain again. You break the chain again. You can recognize each amino acid residue based on the chemical properties. That techniques we call it as a mass spectrometry, nuclear magnetic resonance. All we will work on the next class or coming classes. That's the basic understanding. You have to extract protein, you have to degrade the protein, cleave the protein to study its structure and function, and in terms of the basic fundamental unit. So that comes back, we study a lot of stuff about the fundamentals of the proteins and amino acids. So that's what it brings back into the study of the proteomics. Proteomics is a large scale study of proteomes. You are looking not for a single protein here, you are looking for proteomes. And you take an example of a proteome. A proteome is a set of proteins produced in an organism, system, or biological context. It may refer, for instance, to the proteome of a species, for example, human proteome, human Homo sapiens proteome, or an organ, for example, a cancer tissue proteome, a liver cell proteome, a developmental cell, eye proteome, like that, you can study the proteome. So the proteome is not constant again. It is influenced by environment, and it changes from cell to cell, and it changes over time based on the environmental influence, not only the genome. So that's what a transcriptome, where mRNA codes the proteome, but it is not constant because genome or transcriptomes are static, right? They are the DNA, they don't change much. But the proteome is dynamic, it changes a lot. For example, an example you can take, you study an example here, insect metamorphosis. A caterpillar that goes into a butterfly, and you see that the butterfly has a complex phenome that gives different pigments, different pattern of the pigments. That is basically an outward implicit or the proteome in action, for example, here. And it's a dynamic activity of the proteome that makes a butterfly out of a caterpillar. And the caterpillar may have a genome or transcriptome that may not go that much changes. You can say static genome. The proteome is outward. And you should remember that it's the same species of butterfly influenced by environmental condition. It changes the pigment, proteome level, protein levels different. So basically, that's proteomic analysis, or proteomics helps in studying, example, when and where proteins are expressed, rates of protein production, degradation, and steady state abundance, because you need to have a specific protein. If required in necessity of a process demands, you need to make the protein in that multiple amounts of the protein compared to X and Y protein. Y protein may be multiple. Also, how proteins are modified, post-translational modification, that is important for functionality of the protein. A modification of PTM in a protein X gives you experimentally a Y function rather than an X function. The functionality changes with the modification. And also the movement of the proteins, where they are moving along, they are moving into the nucleus or cytoplasm, or they are in the mitochondria, they are in other part of that, we need to look into that. Then how proteins interact with each other. That's a proteomic in summary, but let's take back into this concept, the involvement of proteins in metabolic pathways. That is our second part of the lecture, that called metabolomics. So is it clear? Proteins, protein structures, fundamentals, concepts, protein modifications, and why the proteome studies. Okay, I will move on to the second part of the lecture. This lecture talks about metabolomics. Metabolomics, again, we go back into the fundamental units. Metabolites, because human body is a very complex, as we studied with the proteins, but at the same time, there are different metabolites are present in the body, depending on the function of that. Liver, spleen, stomach, and then brain, you see a different metabolites that processed and produced, etc. And we will talk about these concepts here in the further slides. So this slide brings back us to the omics studies again. We already know transcriptome, genome, genomics. But coming on to proteome, we studied in the first half of this lecture, and metabolome. For example, metabolome, the proteins again, they kind of, the metabolome gives rise to specific moieties, fundamental units again. They are nothing but building blocks of nucleic acid and proteins. They are sugars, nucleotide, amino acids, and lipids. They produce specific kind of metabolites in the body. Metabolites can influence a phenotype or function that's called as a phenome. In this slide, you can see that starting from the genome, a specific set of the genes, they can produce specific set of transcripts. So again, proteins, again, metabolites. You see that numbers are exponentially growing till this point. For example, a specific set of genes in an organism can produce up to 1 lakh proteins and transcripts. So there is an exponential growth here, but at the same time, the metabolomites, it's kind of only 2,500 metabolites so far found in human genome or human bodies. So you talk about the genes and you talk about metabolites, then you go around into the specimens where you isolate tissues or cells into these first two steps. But when you comes into proteins and metabolites, you have to go into the body fluids, biofluids also. So what is a metabolite? A metabolite is a chemical molecule that is used or created by chemical reactions within the cells or a living organ that are happening within a cell. That's a metabolite. It's a chemical molecule. You should remember that. It's a chemistry all along again. Metabolite is an intermediate or end product of cellular metabolism. It may not be the end product of a specific metabolic pathway or specific metabolism in the body. It may be an intermediate or it may be an end product. We already know that there are two metabolites in the body, the primary metabolites, secondary metabolites. Primary metabolites are components of a basic metabolic pathways. You should understand that primary is important. It's a basic metabolic pathways that are required for life. They are associated with the essential cellular functions such as nutrient assimilation, energy production, and growth or development. These are fundamental. You need primary metabolites. And the secondary metabolites indispensable. They are dispensable. Dispensable and not absolutely required for survival, but they might have some separate functions. For example, a pheromone is an insect social behavior, and pigments are odorants. They are kind of protecting from other grazing from animals. That can be one more. And pigments gives rise to butterflies, a kind of beautiful phenotypes that can be worked. Again, going back into primary metabolites, you're talking about the fundamental units here: amino acid, fatty acid, sugars, and nucleobases. At the end, they give rise to the one of the important blocks of the body. The important components of the body are cellular metabolism: proteins, lipids, carbohydrates, and nucleic acids. Okay, there is an intermediate structures will form. The secondary metabolites, again, they are not of primary importance. They may be dispensable, but they will have again a specific fundamental building blocks. They will have a specific chemical structure. For example, here alkalides, they have a building block of nitrogen. At the end product, nicotine will be formed. Terpenoids, it's a natural terpene, like the different compound secondary metabolites that are also important for some of the functional aspects of the organism or the body. So we talked about metabolites. Coming back to metabolism. Metabolism is a vital process of all living forms, all life forms. You need a metabolism. It's a kind of that makes us alive, and it produces all the things required for our growth and development and energy. It's broadly defined as sum of all biochemical processes maintaining living cells at state of the art. So here in a certain encyclopedia, Kaito encyclopedia, they have studied several organisms and genomes. They could able to see that a specific set of group of genes and genomes, they could able to isolate, confirm that 87,000 chemical reactions are happening in the kind of metabolic processes, and also 16,000 metabolites they identified. So see that there is a vast heterogeneity, vast complexity in this process of metabolism and metabolite production. And we all know that fundamentals: anabolic process, catabolic process. Catabolic process breaks, degrades the molecule, release energy. Anabolic processes from the fundamental units, it makes building blocks, so it makes important macromolecules. Besides that, metabolic processes help it to get rid of the toxic substances in a body. So let's take an example of human metabolome. So there is a complete set of metabolites in the human. You take about it. You want to see and the different aspects of chemical composition of that. There may be exogenous, endogenous molecule, and trans. So looking at here, human metabolome database, they found out that in a body, given a native condition, given a native form of a cell or a body or a human or organism, there will be metabolites that are endogenous to the system. So you're not adding from exogenous. They are already present de novo or present in vivo. They're called endogenous metabolites. It's been found that close to 8,500 metabolites are there in human endogenous metabolites. And here it takes in the concentration of the molecules based on the variety. The concentration may be in femtomolar to molar. And as you look into more, this is endogenous. You look in the external condition here, the four things that may be influenced by environment or maybe exogenous components. For example, drugs, food additives, drug metabolites, toxins, or chemicals. You see that these are exogenous. Again, there is an influence for exogenous metabolites. So human metabolome is diverse. It needs to deal with a lot of chemical reactions, a lot of chemicals, and also make homeostasis of the metabolome. For example, a toxin, you take an example of snake bite. So you take endogenous metabolites level without a snake bite. And the snake bites you, and small amount of the snake venom goes into our system. It completely changes the physiology of the system. So there is a setup one physiology condition, two. That metabolomics, you can study that. Again, you take a drug for a fever, it also gives a condition, changes the physiology. The similar scenario, you can look into a system of food, whatever food you are eating up, that also influences the system. Not only these exogenous, so in summary, on to metabolism. So this is a snapshot of metabolic pathways, right? We have mitochondria, cellular respiration, we have urea cycle, we have cholesterol production, we have pentose and glycolysis pathway. A lot of complex pathways, complex proteins and enzyme, they work in time, they work in space, and they work highly in efficient manner, in a coordinated manner, so that the system moves on. And you see again compartmentalization. One part of the cycle happens in a mitochondria, one happens in cytoplasm, one happens in other cells. So it's a kind of homeostasis. So there is a kind of very nice metabolite interaction and metabolic pathways interaction that makes a homeostasis. It gives an organism to work and run normally. So not only the metabolites in terms of exogenous produced by drugs or produced by an external condition, other than to endogenous metabolites, one of the important concepts we need to understand is called microbiome. In a human body, in a specific given a sex of a human organism, human type, then you look into different parts of the body, for example, oral cavity, nasal cavity, gut or skin, you might find a specific bacteria, and then specific bacteria or fungi may be also existing there. They also have a specific microbial pathway, specific metabolism. They depend on the host, we are humans. They need to survive, they need to go through their own metabolism. In that crosstalk, host pathogen interaction, they might produce a specific metabolites external to the endogenous and based on the community composition, gram negative, gram positive, or fungi or another species that associate. That gives a specific genomes of humans, microbes, and then the microbiome consists of whole set of the genomes, metabolites produced, and that correlates to the phenotype also. It's been now quite obvious that as the environmental condition, your microbiome also influences your phenotype and your other aspects of the development and disease. So coming back to the again definition, metabolomics. Metabolomics, it refers to the study of the metabolome. If you take a metabolome, it refers to complete set of chemical compounds involved in an organism metabolism. It may be intermediates, there may be hormones, signaling molecules, secondary metabolites. And basically a metabolome reflects the state of the cell or an organism or an organ. Study of metabolomics, metabolomics. In functionality of metabolomics, you can study in fundamental aspects, for example, relative differences between biological samples based on the metabolite profile. You're not looking at the genome profile, transcriptome profile, you're not looking at the condition, you're just basically looking at the metabolite profile from sample A to sample B. And also it gives an instant summary of physiological profile of an organism. In terms of the diseases, physiological condition, metabolism maintains the homeostasis. If you want to see that how is our organism doing, you need to go into a metabolome profile. So again, in terms into the advances, it helps in personalized medicine. I may be different from you, you may be different from your friend. So you might need a personalized treatment where the metabolomics can provide a physiological condition of you, or in terms of studying the metabolomics over a time, you can give a personalized medicine, develop a personalized medicine. And also nutrigenomics, based on your nutrition, you can change your nutrition diet, that also influences. In addition, in terms of toxicology studies, you need a metabolomics that influences this outcome of this. So in the present class, I may not go in deep into in terms of the methods used to study metabolomics and proteomics. We will take it in a separate class going into the curriculum. But we talked about exponential growth of level of from genome static genome to proteomics, that is dynamics. Exponentially the number grows up. We already talked about metabolomics, physical and chemical composition of protein and metabolomics. You should remember that you cannot use a PCR or microarrays to study the proteomics, but you need to use a specific kind of mass spectrometry, specific kind of NMR techniques that works on a chemical structure of each amino acid residue. That we will talk in further classes. But in a kind of brief, the methods used are mass spectrometry, nuclear magnetic resonance, HPLC, high performance liquid chromatography, or high pressure liquid chromatography. You can use LCMS, combination of liquid chromatography and mass spectrometry, or you can use gas chromatography and mass spectrometry together. You study this kind of understanding the fundamentals of amino acid residues, protein structure. And also one important aspect is crystallography. You can crystallize the protein to work on its three-dimensional, native three-dimensional structure, but crystallography is costly. So you go for simulations first, then you understand the simulation and functionality process going fine. then you can go on to crystallize the protein. But a lot of bioinformatic tools are available to study each and every concept of it that we will discuss in further class. So in terms of disease network, how the metabolomics helps. For example, you take a network mapping. For example, you are looking at the physiological condition of amino acid sugar metabolism, glycolysis, TCA, amino acid metabolism in a condition that is in a wild type condition that is consists of a patient or a person in a wild type situation. You will have, for example, all are gray, all are in a native endogenous levels, all reactions are happening, network is established. But you go into a disease scenario, you develop into a kind of diabetic patient, or you have a high cholesterol levels, you get into some other heart problems, then the metabolomic profile, the network profile changes. For example, red is increased amounts of these kind of specific set of network molecules or metabolites that have been increased here. Or green may be a decreased condition. You can get a network of that, and a gray is a condition that doesn't change. So that gives you a black and white scenario of a disease condition, normal condition. Then you can go ahead and plan with the drug targets treatment plans. That is one of the concept of helping disease in a metabolomics. So again, these molecules in a native condition, for example, any amino acid or any nucleic acid, or for example, any antibody produced in a cell, they work as biomarkers. What are biomarkers means? A specific disease condition that may exist. You may not need to go ahead with taking whole network. You may go ahead with analyzing specific set of biomarkers where the condition or level of biomarkers can help you that a disease is exaggerated or it's going normal or not. That we will study in the next 2 slides and then stop the presentation. So biomarkers, they are naturally occurring molecule, a kind of gene or characteristic by which a particular pathological or physiological process, disease, etc. can be identified. So it gives an indication, marker for a specific biological function. A biomarker that may predict aggressive disease recurrence in liver transplant patients or liver transplant recipients. A person who is gone for liver transplantation, you might have a biomarker levels that go ahead aggressiveness that tells about disease reoccurrence. Or you call it as a biomarker in otherwise. A biological marker generally refers to measurable indicator of some biological state or condition. That in a normal condition, physiological condition. The condition may be at the level of your studying biomarkers at the level of a characteristic, that is at the level of molecular level. It may be anatomical level, it may be physiological, or it may be biochemical. So that you study as a biomarker. For example, the biomarkers, potential biomarkers, including proteins and protein fragments, metabolites, carbohydrate biomarkers, genomic biomarkers, RNA and DNA, cellular biomarkers, a kind of body fluid, cell pellet that consistently shows a specific biomarker, and many imaging biomarker you can use to study. Coming back to the talk for today, proteomics and metabolomics, we just briefly introduced to the concepts, 2 concepts, and how they will functionally important for studying disease models. And that giving into a conclusive that we come to the conclusion of Unit 1 for the genomics and proteomics. We already introduced genomics, proteomics, metabolomics, and transcriptomics. All the concepts for the basic level are covered, but we take up more into the advanced system. We go into the curriculum, we take each concept, we take each method, for example, microarray, for example, NMR, for example, then how do you sequence a genome, how do you study a complete genome, then in terms of metabolic profiles, we will further go ahead into the further class. Okay, I will stop the recording now. Thank you for your attendance and thank you for your patience and attention.
Thank you, sir.